Hello and welcome, everyone. My name is Kristen Moore, and I am the Webcast producer at LCGC and your moderator for today's live broadcast of Breakthrough Forensic and Drug Testing Bottlenecks in Seconds with Mass Spec Solutions. We are delighted to bring you this educational web seminar presented by LCGC and sponsored by PerkinElmer Incorporated. PerkinElmer is a global leader focused on making a difference for better human health and a safer environment. They create solutions that help detect, prevent, and cure threats to human and environmental health. PerkinElmer is dedicated to the quality and sustainability of the environment. With their analytical instrumentation and leading laboratory services, they focus on improving the integrity and safety of the world we live in. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the red Q&A widget at the bottom of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. Today we are very pleased to be joined by Andrew Tyler, PhD, Global Field Technical Leader for Mass Spectrometry at PerkinElmer. Dr. Tyler has worked with and around mass spectrometers and chromatographs and their application to small molecule structural challenges all of his professional life. He received his PhD in 1982 from the University of Manchester Institute of Science and Technology in the UK. Dr. Tyler has worked with diverse environments and applications, including high-throughput synthetic characterization, in vivo and in vitro, PK/ADME, metabolite identification, metabolomics, and lipidomics. Thank you for joining us today. Andrew, please go ahead and get us started.
Thank you, Kristen. Thank you, everybody, for joining us today for our webcast. As Kristen said, my name's Andrew Tyler, and my job involves providing mass spec technical support to PerkinElmer's field scientists and engineers, as well as to our customers. Today, I'd like to share with you some information on the work we have been doing at PerkinElmer in the field of forensic sciences. At PerkinElmer, we recognize that modern forensics presents a somewhat daunting challenge with a broad, diverse array of fields in which to study. There are many topics that encompass forensics today, including blood measurements, ballistics, workplace drug screening, and pain management, and all of these fall under this broad umbrella of forensics. And we understand that the time and cost challenges of working in this arena, combined with the challenge of providing robust and high-quality data that may even need to stand up in court proceedings, presents a major challenge for people working in this area. And we're very mindful of these factors as we have tried to develop methodologies, equipment, and protocols that will make life easier for people working in this arena. As I've already mentioned, forensics is a term that can encompass many areas of study, and I went online and found a definition that, as you can see on the top of the slide, it's a very broad definition. And interpretation is somewhat subjective to begin with. For example, if vegetable and fruit produce is fraudulently labeled as being organic, is that in fact a forensic application? However, for today, we'll consider forensic science as falling into four main application areas. And today we're going to focus on the first two of these, forensic evidence, and workplace drug analysis. Within these two arenas, there are subdivisions that we are going to present examples of work around these. Illicit drug screening, gunshot residue analysis, and fiber analysis will be examples that we'll discuss from the arena of forensic evidence. And within workplace drug analysis, which is a somewhat regulated environment, we'll discuss the challenges of working in this arena and present some new workflow ideas that may assist for those of you working in that landscape. There are many different tasks that fall to a modern forensics laboratory. Some of these tasks might involve molecules with very high molecular weights, particularly DNA and larger proteins. In our mass spectrometry applications, we're focused on the low molecular weight molecules, and today we will not be talking at all about the higher molecular weight problems. So now we're going to turn to consider specific examples, and first of all, we're going to talk about illicit drug screening. Illicit drug screening is an area that is ever evolving. Novel synthetic compounds that have not been the subject of any legislation are emerging every week. Raids by law enforcement generate huge numbers of samples that require characterization to just figure out what's actually in them. I recently spoke with a director from a law enforcement agency whose laboratory received somewhere in the region of 7,000 samples in one day as a result of raids being conducted in that area, and these all have to be analyzed and considered. Chromatographic methods can be time-consuming to develop, to execute, and also expensive in terms of materials required, as well as the equipment. The cost of solvents, for those of you who work in this area, as you know, are quite expensive and require disposal after their use. So here is what we consider to be a fairly routine type of protocol that's followed in many labs doing this type of work and will be familiar to some of you. And this involves then some sort of extraction procedure, pipetting, dispensing, putting samples onto instrumentation and running them, collecting data, analyzing the data, and confirming results. And we consider that this sort of thing, that 25 minutes is a fairly reasonable estimate of the labor that goes into something of that type in a routine situation. So 25 minutes per sample is our estimate. Now, if you contrast that to the type of work that you can do using what we call direct sample analysis, or DSA, that workflow is dramatically reduced in terms of time and resources. Small aliquots, typically 5 microliters of a solution or a small amount of powder, can be introduced into the mass spectrometer and ionized and analyzed very rapidly. In DSA, samples are ionized by interaction with a beam of hot ionized nitrogen gas, and the data from the sample exposed is collected in roughly 25 to 30 seconds. On the right-hand side of this slide, you can see a picture of 2 different ways of introducing samples. Metal grid system is available for depositing liquids, or glass capillaries can be used for adding powders and doing direct analysis of sample powders. So here a 25-minute workflow has been replaced by a workflow that requires roughly 25 seconds to execute. Here we're looking at a picture of the DSA, the direct sample analysis ion source. And this is the device then that fits onto the front of the mass spectrometer as shown in the insert picture at bottom right of this slide. And this was designed with ease of operation and change between LC and DSA mode in mind. All of the gas and electrical connections for this device are plumbed in for automatic connection There is a reagent line inlet as shown on top of the device, which allows you to add standards and modifiers as solutions directly from an infusion pump or other liquid device. And later on, we'll see an example of the utility of this and why you would want to actually have that available. And so this mounts on the front of the mass spectrometer to change over from liquid chromatography mode to DSA mode. Inexperienced hand takes a couple of minutes, but even for a novice doing this for the first time, this is no more than a 5-minute changeover. It's very simple and very straightforward to do that. And so this device is sitting on the front end of our mass spectrometer. It's a time-of-flight device, which we call the AxION TOF2. And the AxION TOF2 is a high-resolution mass spectrometer. And for those of you not familiar with high-resolution mass spectrometry, here's one slide just to talk about the strengths of high-resolution work and how it helps in this field. Many of you are probably familiar with triple quadrupole instruments, which have low resolution or unit mass resolution capabilities. And so this slide is intended to give a little insight into why high resolution is a helpful technique. On this slide, as you progress in the figures from left to right, this is intended to show the impact of increasing mass resolution on signals that are detected. And the key factors here are that as the resolution increases, the confidence in the mass assignment of a recorded peak goes up, so that you can assign masses not just to unit mass, but now to 4 significant figures with a high degree of confidence that the mass measurement is accurate. And what this enables you to do is that at high resolution is that you can assign a composition of the ion based on the mass alone. So in this example, then on the left-hand side, you see that there are multiple formulae that you can postulate, all of which are consistent with the measured mass accuracy. And as the resolution goes up, so the confidence becomes higher until in the right-hand panel you have sufficient confidence in the number that you can reduce the number of possible compositions to a single or unique composition. The second factor that is enabled by this technology is the precision of the isotope peak measurements. So in the right-hand side, you can clearly see that there is an isotope pattern there. The second peak above the most intense peak is consistent with the presence of a single chlorine atom in that signal, and the formula underneath indicates the presence of that chlorine. So those isotope ratio intensities can be used as additional information beyond that of just the mass to infer confidence in the assignment that's been placed on this composition. So we're using both the accuracy of the mass measurement and the stable isotope pattern that's an inherent feature of every molecule that one measures. So enough about high-resolution mass spectrometry for now. Here is a picture of the DSA source in action. As you can see on the left-hand side, there's a metal grid system available for aliquoting, pipetting liquids onto the grid. In the central picture is a glass capillary. Powders can be inserted into the top of this capillary. And on the right-hand side is a snapshot of the software that runs this device, the DSA controller. You can run up to 13 grid samples at a time. There's also a list there of targeted molecules, which we'll speak more of in a minute, but you can enter a list of named molecules and their compositions as targets of any experiment. The software that's used for processing the data is known as AxION Solo. So Solo is the tool that we use for processing data in rapid fashion. And this slide shows you an overview of a typical observation panel in Solo. So in the upper left there's a grid showing samples that are run, and the colors in that grid reflect the detection or otherwise of molecules that are in the sample list below that. So in the list below that is a list of molecules and their compositions that one is searching for. So this is the target list for the experiment. In the upper right-hand panel is the isotope pattern for a given molecule, both detected and calculated. So this is the point we were just making about precision of isotope patterns and using them to infer confidence in the assignment. And in the lower panel is an annotated list of substance peaks as were detected in the DSA experiment. So this enables then very rapid manual screening through the data to see what was seen of an expected targeted list and what was not across a whole batch of samples. In the following slide, this is an example of the reporting features coming out of that software, and this can be used for reporting to notebooks or electronic notebooks or whatever reporting process is required in a laboratory. In the upper panel there are spectra showing the detected peaks and isotope ratios, and below that is a list of what is found, what is not found from the target list, together with the measured and expected masses and reflection of the precision of those results. We're now going to talk briefly about a real-life case study. This is all very well with regard to standards and so on, but when it comes to the real world, what really happens? So here, one of our scientists was working in a laboratory, in a real forensics laboratory, and was presented by the collaborator with, as it turned out, 369 samples that were a variety of seized pills, vials, powders, and a few urine samples, and presented to our scientist as unknowns. And he was given the task of working through these and seeing what they could really do with the machine. The upshot of this was that of the 369 samples, 53 unique targeted substances were detected at a runtime of around 25 seconds per sample. And so to actually collect the data and to review it using Solo was around 2.5 hours of work. So when the sample preparation time and a presentation to the machine is considered, all of this work took around half a day to get through. So this was considered by the collaborator a real positive for their lab, and the data were confirmed by the methods that the lab was running in parallel to this, but of course over a much longer time frame to do that work. Here is the list of some of the molecules that were detected in that study, and a description of why they are there, what their psychoactive response is, together with their formulae and the expected and theoretical masses of this. And the results here are presented in terms of accuracy of the result. In high-resolution work, data are typically presented in parts per million deviance from the expected mass. So this is not a concentration parts per million. This is a mass measurement parts per million. And on this type of instrumentation, typically results within plus or minus 5 parts per million are considered to be consistent with the performance that one would expect from this type of equipment. So the measured masses are very close to what they're expected to be, and this gives confidence that we are in fact assigning the correct composition to the measured mass. On this slide, we're looking at the Solo presentation from a partial list of those, and what one can see on the left-hand side is that the cocaine peaks are being labeled as blue, the measured masses are shown, and the peaks annotated. And the list on the lower left shows which species are measured and the mass deviation. The absence of a number in that list merely informs you right away that they were not detected in that particular sample. Moving on then now to look at the AxION Solo setup. Here is a list of species. These lists are very easy to create. You merely enter the name of a molecule, what you want to identify your target as, and the formula that's expected for that. And from that, the software will calculate the masses, and it will also calculate the isotope pattern. And these lists then can be saved, they can be edited, they can be modified according to the user's needs. And so you can have one master list, or you can have a list of just amphetamines, for example, whatever it is that's consistent with your workflow. So this is very easy to set up, and once it's established, you can run it forever. This is another software tool that we have. It's called ECID. And ECID then allows interrogation of these isotope patterns and masses and determines a fit of the empirical data with calculated patterns for each composition proposed. And so in the box of the list of compositions, each of those can be interrogated, and the nature of the fit with the expected data can be compared. In addition, this tool will allow you to compare anticipated results with databases. These can be public databases, or they can be ones that are established in-house according to the needs of the customer. And what's happening here is then a particular result is being searched against the PubChem database, and this is an example then of one molecule within that database whose elemental composition is consistent with the detected data. And so this is a tool that, when you have unknowns, will allow you to go out and start to get some idea of possible fits with the measured data. On the AxION machine, one can induce fragmentation of molecules in many cases, and this tool will also allow you then to predict possible compositions for those fragment ions based on what the composition of the starting ion was. And where this becomes a useful feature is shown on the next slide then, where you can see in cocaine, there's a number of fragment ions which are circled. The one on the right-hand side, 304, is the molecular peak. The remaining circled ions are fragments of cocaine. And those species can then be added to a database. And so in this solo snapshot here, not only is cocaine entered as a target, but also the fragment ions in addition are entered. And the virtue to this is then that if you're seeing a molecular peak of cocaine, you should be able to see the same fragment ions consistently across the run. And so now you have not just one species as confirmatory for cocaine, but in this particular case, there are 4 different masses. All of them are consistent with cocaine, and you should expect to see that. So this is adding a layer of confidence in the assignment beyond just the molecular peak being detected. Here's an example then of reporting coming out of this experiment, and you can see on the right-hand side that there's a list there of the detected cocaine fragment ions, as well as the mass accuracies with them. And again, these are being shown as being consistent within the 5 ppm that we would hope to see. Okay, moving on, we're going to talk a little bit about, still in forensic analysis, but now we're going to have a look at analysis of gunshots and fibers. So this is a field where scientists who work in this field are always seeking new ways to link gunshot residue and powders with established profiles of known materials. And this is a particularly challenging area to work in. And one of our scientists was then presented with 2 different types of gunshot residue, he was given the identification of RP for one of them and Winchester for the second, and was asked to see if he could determine any differences between these residues. This is a case where the ability to enter a modification agent via the reagent inlet line I mentioned earlier in the talk was very useful, as you will see when we talk about the data here. So one of the components that is expected or potentially expected as being present is nitroglycerin. And nitroglycerin is a molecule that is difficult to ionize with DSA. It's difficult to ionize with most mass spectrometry methods. However, what our scientist was able to determine was that if he added a small amount of dichloromethane in a methanol solution and infused this into the machine through the reagent inlet, then he was able to generate an adduct ion in the negative ion mode, an adduct ion of nitroglycerin with a chloride ion, so M plus chloride. And in the expansion box on the right-hand side of the slide, you can see this is clearly a confirmed identification. The isotope pattern of chlorine is seen as expected, and the mass accuracy of the measurement here is consistent to less than 1 part per million of what was expected. So this is a way then of using the flexibility of the technology to measure a species that was otherwise unavailable to us, and he was able to detect nitroglycerin in this fashion in the RP gunshot residue. When he looked at the Winchester gunshot residue that was provided, he was not able to see any presence of that molecule here. It's essentially a blank-looking. Nitrate and nitrite are everywhere around explosives, so there's no surprise there. But he was not able to see it in that particular case. Switching over to the positive ion mode now, so this is the same sample preparation, but now being ionized in the opposite polarity on the mass spectrometer. The molecules ethyl and methyl centralite, which are very common components of gunshot powder, were clearly seen as being present in that case. Moving on to talk about fiber analysis. Fibers come from many different places, of course, carpets, furniture, clothing, many cases where such an analysis is desired at a crime scene. And fiber analysis is performed today with many different technologies and spectrophotometric methods, as well as TLC, HPLC, and with UV detection. We can also contribute to this field using HPLC coupled to a time-of-flight mass spectrometer, which gives a more specific identification of which components are present. And as I will show you, you can also potentially do quantitative analysis of this type of thing. So here's an example of 3 anionic dyes that are common in fibers that are amenable to analysis by LCMS and negative ion detection. Here's a chromatographic method for the separation of these 3 components, and the precision of the mass measurement is within 2 parts per million of the expected mass. So we're highly confident that we have the correct identification here for these components. These can be quantified. So here's an example then where those 3 components have been run and a standard curve generated from those solutions. And as you can see in the figure, there's excellent linearity over the analyzed range for all 3 of the components. So this technique then was applied to some real fibers which were provided to the applications lab, which were extracted and appropriately diluted. And using the calibration curve approach that I previously showed you, the scientist was able then to provide concentration data, and on an N of 5, these were all to within plus or minus 15% standard deviation. You'll notice in the chromatographic figures that one of these fibers has the 3 components present, whereas the second fiber only has 2. So the scientist was able to distinguish between the two fibers just based on the pigments that were extracted from these fibers. So to give a quick summary then of the work that we've done on forensics, I've shown you the DSA, the direct sample analysis approach, and how this is a tool that can enable far, far shorter analysis times and assist the workflow in that manner. It's an excellent tool if you're confronted with lots and lots of samples that require a qualitative identification comparing against a targeted list. You can move through a lot of samples very rapidly using that tool. Screening and confirmation using the power of high-resolution mass spec to obtain accurate mass measurements allows us to give confident compositional assignments, and the information provided by stable isotope pattern measurements allows us to further enhance the confidence in the data. As we mentioned, the whole system is designed for flexibility and easy change between the DSA approach and an LC-MS approach, and changeover from one to the other is accomplished within a few minutes if necessary. Okay, so moving on to the second arena that we want to talk about today, we're going to turn our attention now to workplace drug screening. As many of you may well be aware if you've changed jobs in recent history, this is a very common feature of hiring practices in the United States, and routine screening practice for many industries, for example, people who work in shipping and delivery business are often subject to random workplace testing. It's a common practice to use immunoassays as the primary screen in these types of assays, and then follow up where warranted, where a positive result in those initial screens suggests that a more specific follow-up is required. Immunoassays are wonderful tools for high throughput, but they do have some issues with regard to cross-reactivity. Many of the drugs that are being screened for, of course, have structural features in common, and they're potentially, therefore, are liable to give false positives in an immunoscreening approach. Confirmatory testing is often done using, typically in many labs, a GCMS approach, where runtime and mass spectrometry, so retention time and mass spectrometry are the features that are used in the confirmatory testing. In more recent times, the federal government in North America has mandated testing for people in certain occupations, and in the U.S. this is administered by what is known as the Substance Abuse and Mental Health Services Administration, or SAMHSA, who are responsible for writing the regulations for this type of work. Historically, this was regulated by what was known as NIDA and was known as the NIDA panel. And while that is a somewhat out-of-date term, you'll still see it referred to in many cases. There are 11 drugs in this screening panel, and each one has its own cutoff level. Here's a list of the drugs that are screened for here, and the acronyms that we'll be using today as we talk about these. So here is a list again of the drugs and the class of compound from which they come. On the right-hand side of this table, we're showing the molecular weights expected for these drugs and their formulae, and also the screening cutoffs as mandated in the panel regulations. As you can see, there's quite a range of concentrations here, and so our task is to make sure that we're able to detect each of these compounds with sufficient sensitivity to meet the mandated cutoff levels. Here are the experimental conditions that were employed for this study. This is showing us the chromatographic conditions that we used. The components of the buffers here are fairly standard for HPLC buffers in mass spectrometry, where one of the prerequisites is that all of the buffer components are volatile and are not going to clog up the machine and negatively impact sensitivity. One notable thing to point out here is that the diverter valve on the machine is employed. So what this allows one to do is to divert the LC effluent away from the mass spectrometer to waste on a time-controlled manner. And in this experiment, for the first 1.8 minutes of the run, that was done. And the reason for this is that urine tends to be a very salty matrix from which to work. And so by doing this, the salt, which is coming out in the early elution volume of the run, is diverted to waste, and it's not clogging up the mass spectrometer. And the result of this is that we have a cleaner and a more robust method as a consequence of doing so. The column that's employed here is what Brownlee call an SPP column. SPP stands for superficially porous particle. And this is a matrix that allows a favorable trade-off between flow rate and back pressure, which enables one to then up the flow rate relative to the capacity of the column and gives a consequent improvement in the chromatography compared to a standard material column. On the mass spectrometry side, here's a list of the values. The important thing to understand on this slide is that the mass range is being set to cover the range appropriate for all of the molecules that we expect to see in that panel. And all those ions are being collected all the time. There's no MRM. If you're a triple quad person, you're familiar with establishing MRM methods to analyze these. There is no need to do so on a time-of-flight machine, which is looking at all the ions all the time across a selected mass range. And so method development is fairly straightforward. The acquisition rate here is 2 spectra per second. If you're used to working with MRMs, that seems like a ludicrously low number. But again, remember that the entire mass range is being collected with every single acquisition. And so on a method here where the chromatographic peaks are 5 or 6 seconds wide, there's a regulatory requirement to have at least 10 points across the peak as per the regulations for this. And so that rate is consistent with meeting that demand while collecting sufficient ions to give statistically valid peaks and improving reproducibility of the method. So we're getting at least 10 spectra across each chromatographic peak, but we don't have to have the machine flipping around between MRMs to accomplish that goal. If we move on to look at sample preparation, it's very straightforward. This is a demo situation where urine samples are being spiked with aliquots of all 11 panel drugs. And in addition, we're adding deuterated internal standards of 10 of those 11. So there's one exception where material wasn't available. And that's then used for quantitation purposes of each material. Those samples are diluted 1-to-1 in water, so in essence it's a very straightforward preparation. It's addition of standards and along with a 1-to-1 dilution. Here's a chromatographic representation of the results of that. It's about a 10-minute method to cover all the components in the panel. And what's being shown here is extracted ion chromatograms, or EICs, is the acronym being used here. And so each of those traces represents the abundance of a mass of one of the target molecules, or the mass of the target ions, across the entire run. And so we're confident that each peak is as it is labeled across the run, and you can see that each of those materials has been detected successfully in that run. Here's an extracted ion chromatogram for the drug MAMP, as well as the internal standard for that material. And so as you would expect for an isotopically labeled peak, the retention time is exactly the same as for the drug itself. And then the ratio of abundance of those two peaks then allows you to quantify the MAMP. So it's a very straightforward process of quantifying that material. Using the AxION Solo software to look at this panel, processing the data I just showed you, there are 11 drugs and 10 internal standard substances in our target list here, as you can see on the lower left-hand panel. Each of those materials is shown as being present based on the mass difference between expected and measured mass being shown there. And so there's a very rapid tool for interrogating the data and confirming that each of those molecules has been observed. In terms of standard deviation on the data, here's a list of that. You'll notice on the right-hand side we've included the limit of quantitation as assessed through development of these methods. And the key part here is that the numbers in the right-hand column are all significantly lower than the numbers in the cutoff level. In other words, we are able to detect and quantify each of those materials below the level that would be required in a regulated laboratory. One of the other criteria for the panel regulations is an assessment of carryover. The method mandates that the carryover measured from a run containing a highest concentration standard to a blank injected immediately afterwards, that the carryover percentage should be less than 10%. You can see from these data that all of these numbers are significantly lower than that. All of them are below 1%, and some of them are not even measured at all. So our method is well within bounds of the regulation with regard to carryover from one sample to the next. Here's a table of linearity and linearity coefficients that were determined here, and over 3 logs of concentration range typically here. The only one that isn't is the THC metabolite. All of these are giving better than 2 nines linearity across the experiment based on an N of 5 for each point. So we have very good linearity in the method as well. Here's a final example of using the isotope profile for additional confirmation that the molecule is the right one. This is MDEA. So we have an exact mass that's better than 2 ppm in terms of the measurement precision of the mass. The isotope pattern, the first isotope peak, the measured abundance is within 0.5% of the expected abundance. So we are, again, using that information to reconfirm that we're assigning the correct drug to the measured peak. So a quick summary then on the work we've done in workplace drug screening. We've shown that we can use a time-of-flight mass spectrometer coupled with HPLC to provide an accurate solution for quantitation and confirmation for workplace drug screening. Methods development on a TOF system is considerably easier than one would encounter on a traditional triple quad method used for this type of work. LC development time, of course, is the same regardless of the type of mass spectrometer on the end. But setting up the mass spectrometer is significantly easier on a time-of-flight system. The final advantage here is the ability to actually go back and reinterrogate data, which is something we haven't talked about and not necessary in a regulated setting, but the ability to go back to these data sets at a later date and interrogate for drugs that were not suspected in the original screening or metabolites of drugs that had not been anticipated. With TOF data, if you have the raw data, you can always do that. Whereas if you have MRM data, then you're restricted always to the species that you had on your target list to begin with. So there is an additional potential benefit there. If anybody ever comes back and says, did you see evidence for this? Then you can reinterrogate the data as opposed to having to go back, develop an MRM, and then rerun the samples in essence. So in closing, I just wanted to make you aware of resources that PerkinElmer are providing to community interested in forensics applications. Here's a link and a web page that is focused on forensic work and drug analysis. And this is a great starting point if you're looking for what resources are available at PerkinElmer to help in forensic work. So in conclusion, I'd like to thank you all for joining us today for our webinar and acknowledging the contribution of several members of the PerkinElmer Mass Spec team. Without their hard work, I would not be in a position to sit here and talk with you today about this work. So I extend my acknowledgments to my colleagues for their help. I'd like to thank you all very much for joining us today, for your time and attention, and we will be glad to answer any additional questions that you have at this point. Thank you very much.
Thank you, Andrew, for that informative presentation. Before we begin the question and answer session, I would like to remind our audience about how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the red Q&A widget at the bottom of the presentation window. We'll try to answer as many questions as we can. Our first question came in and is, how can I analyze intact proteins on the DSA?
So the answer to that is no, we cannot. So that the DSA functions with molecules that have some degree of volatility to them. So it's therefore restricted to small molecules, and proteins, intact proteins, are going to be much too heavy to have that sort of volatility. So the answer to that is no.
Thank you, Andrew. Okay, let's pull up our next question. How much fiber, length, or weight was required for the dye analysis discussed?
It's a very good question. So that study was done in collaboration with a customer who actually had done the extractions themselves in what they described as a proprietary process. And we were shipped the liquid extracts to do the analysis. So I do not know how much fiber was actually used in the course of that extraction. So I'm sorry, but I can't give a definitive answer to that question.
Okay, thank you, Andrew. Our next question came in. This looks like it's a two-part question. If there is no sample cleanup for urine samples with 1-to-1 dilution with water, wouldn't that affect the performance of the mass spec down the road? What is the maintenance related to DSA compared to traditional sample cleanup like SPE or LLE? And I can repeat the second question if you need me to, Andrew.
Sure. So let's field the first of those two parts then to begin with, which is really related to contamination of the instrument. And this is a great question, and I thank the person who asked it. So most of you on this call today, I'm sure, have worked or do work with urine samples on a regular basis. And we're all, therefore, familiar with the issue of potential of salt contamination on the front of our mass spectrometer, whether we're doing electrospray or APCI or any of the API techniques. Is there a possibility of some sort of issue? Yes, absolutely. The one thing, however, that I would remark here is that because the actual volume of urine that's being deposited per sample on the DSA stage is really quite small, that we actually don't get that issue in the course of a particular experiment. So is there potential for it to be an issue in a longer term? Yes, of course there is. Is it an issue in terms of the context of one particular study? No, we have not seen that to be an issue. With regard to the contamination of the machine, then the interface where the ions are entering the mass spec is the place where, if one sees contamination, that's where it's going to happen. The instrument is designed so that interface can be removed without having to vent the instrument in order to clean it, and that the capillary design there can either be replaced with a second one for maximum uptime, or it can be taken out and cleaned and sonicated just as on anybody else's mass spectrometer, depending on the operational workflow that works in your lab. So that was the first question. I think the second question was regard to the maintenance then. So I've sort of answered that, I think. Relative to techniques like SPE, it's hard to compare. SPE, of course, removes most of the salt to begin with. So you would expect that the maintenance on the back end is going to be reduced because of that. But then you've front-loaded the effort on the front end with the costs and supplies and so on of doing the SPE. So it's really a matter of individual workflow and what works for your particular application. So there are trades and compensations with all of these, but I wouldn't want to give a categorical answer one way or the other. It's very sample and application dependent.
Thank you, Andrew. Okay, our next question is, how much more time is required for a maintenance for a TOF versus a MSMS system? And is there a significant difference?
So in terms of system maintenance then, for a time-of-flight machine, the biggest issue that traditionally is sort of appreciated for a time-of-flight machine, and when the questioner is asking with respect to an MSMS, I presume that we're considering a triple quadrupole as the comparator here. So the one thing about TOFs, those of you who have worked with TOFs are aware of this already. For those of you who are not, that one of the issues with TOFs is that the detector technology on a TOF is significantly different to the detector technology on a quadrupole. And if the instrument has to be vented for any purpose in terms of maintenance, then there's a significant downtime while the instrument is pumped down to an operating pressure after that maintenance. And if you don't do that, then there can be issues in terms of the performance of the detector following the maintenance. So that's the issue that's most commonly, if you point at TOF and say there's an issue, that's the major issue, I think, that you would encounter. Now, on this machine, we're well aware that that's an issue, and what's happened, as I mentioned previously, with regard to maintenance and with regard to the salt from DSA, the instrument has been designed with that in mind. So there is a shutter capillary type system, so that when the part that gets dirty requires cleaning, or the part that gets dirty most frequently, you can remove that from the instrument. The shutter closes off the vacuum system, so the instrument does not have to be pumped down, does not have to be vented in order to perform that routine maintenance. So when you then reinstall the front end, the act of reinstallation raises the capillary, and the instrument's then ready to go and have a performance check, and you're not waiting for hours for the pump down. Now, of course, if you do have to vent the system for some internal parts, all machines require maintenance on some sort of occasion, then you do still have to be careful with the pump down of a TOF versus a triple quad. That's the same on anybody's TOF machine. There's no exception to that. So that's the one thing that you would point to, I think, as the thing to be considered. And hopefully the design of this instrument has taken that into consideration on a routine basis. So the front ends are the same, whether it's a TOF or a triple quad from any particular vendor. And so there are no significant differences in with respect to the front end of the machine. It's really all about the back end and are there problems. And I think we've just addressed those with the first part of the answer.
Great. Thank you, Andrew. Our next question is, can DSA interface to any other vendor's mass specs?
Okay, that's a very good question. Those of you who are familiar with this type of technology will see and probably know of solutions from other vendors. So we like to claim that the DSA is the only ambient ionization source that you can buy today that's integrated into the same vendor's mass spectrometry product. So if you acquire a similar type of device from anybody else, you're going to have a two-vendor solution, and all the issues that might arise from that in terms of interfacing and to origins of software and so on. So we've maintained the DSA as a PerkinElmer product with the PerkinElmer machine, and we have not ventured out to distribute through other mass spectrometry producers.
Thank you, Andrew. Our next question is, what do you do about matrix interferences in blood and urine? Is it a problem?
So I think everybody on the call is going to be familiar with the concept of matrix interferences and the potential contribution from extraneous materials with the ionization of what you're trying to see. And clearly in any biological matrix, the more complicated the matrix you start with, the more potential there is for that type of issue to arise. So I'm not going to tell anybody that it's not a problem. It's very much dependent upon the matrix that you're working with. The virtue to the DSA is, as you heard earlier, it's very quick and easy to determine whether samples are working or not. Even if it's not the final method you want to run, just making an assessment of can I see something from this particular sample is quick and easy to do that. So you have to consider your workflow, and the workflow with this type of system is a little different to what people are familiar with. If you have a project where you determine that there isn't a matrix interference problem, then clearly there is still a place for SPE and other cleanup technologies. Even if you have to do that, bear in mind that the actual analytical phase of the experiment is still significantly reduced in terms of time and materials at the analytical level, even if you have to do that. So I'm not here to tell you that this is a replacement for all of the workup that you always have to do, far from it. But even if you still have to do workup for your samples, there's still a significant potential for time and cost savings with the DSA technology.
Thank you, Andrew. And it looks like we have time for one more question. Can you quantitate with the DSA?
Okay, so let's take this as sort of a little bit of an extension of the previous question. Are there matrix interferences? Do they interfere with performance? And by extension, would that interfere if you were trying to quantify? And the answer to that, of course, is yes, absolutely yes. There is a scope for interference. Now, what I will tell you, however, is that if you have available internal standards available to work with the DSA, and you can incorporate those standards into your workflow, then it is perfectly reasonable to quantify over a certain dynamic range with the DSA. We've done this with, I'll give you one example. We don't have the data today, but looking at caffeine in caffeinated beverages and assessing the concentration of caffeine. If you can buy commercially a carbon-13 labeled, stable isotope labeled version of caffeine, which will act as an internal standard, just as the same molecule would work in an LC-MS approach as well. So in that respect, it's no different. It's like a one-point calibration. You can ratio between your known and your unknown and quantify that way. And if you do that and set up the experiment appropriately, you can get quantitation data that's quite in alignment with what you get on the same samples done through an existing LC-MS methodology. So the answer is a qualified yes. Like any other mass spec technique, DSA is not inherently quantitative, but it is quite possible with appropriate experiment design to do quantitation with the DSA.
Thank you, Andrew, for those insightful answers and educational presentation today. Unfortunately, we are out of time. Thanks to all who joined us and participated by submitting questions for our expert during today's event. I would also like to thank our sponsor, PerkinElmer, for making today's educational webcast possible. Please note this webcast will be posted for on-demand viewing through October of 2014. You will receive an email from LCGC alerting you when it is available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.